Thanks for introduction. I'm Senying Lai. I come from Fudan University, and today's my topic is about the enterotypes of the human gut microbiome. And let's begin by introducing some related background of my work. And microorganisms are everywhere in our world, and they also present in our human body. And the number of microbial cells is approximately 10 times the that of human cells. And the number of microbial genes is also 100 times the that of human genes. So the human gut microbiome is also called the human second genome. They can take involved in every stage of the human life development, so they are very important. And the gut microbiome is primarily composed of the bacteria, virus, and the fungi. But the previous study most focused on the bacteria and the virus, but forgot the role of fungi in the human gut. Although the fungal composition comprises less than 1% of the total microbiome in the human gut, but they still play pivotal roles in the human gut. The fungi and the bacteria in the gut can have extensive interactions as they share the same microhabitat. And their interactions are also associated with the human health. For example, the fungi-bacterial co-infection could increase the pro-inflammatory cytokines and thus cause the human disease. And the fungi are linked to a wide range of human diseases. Previous studies have reported the alterations in the human gut composition have been found in the multiple human diseases such as the oral cancer, IBD, IBS, and CRC. And the enterotype is an effective method to stratify population based on the human gut microbiome and can provide a global overview of the structure of human gut microbiome. And the previous study has consistently identified the bacterial enterotypes across different studies, which are independent of geography and gender. So this raises a question that whether there exists a fungal enterotype-like structures in the human gut, and how is the landscape of the human gut microbiome. So in our study, we collect more than 3,000 ITI samples from the 16 cohorts across 3 continents, including Asia, North America, and Europe. And from this photo, we can see that the sequencing depth of most of the cohorts is sufficient to capture the full diversity of the fungal composition. And when we combine the whole samples, we can find that the number of observed fungal genera are still below the saturation. So this means that the further in the sample size is needed to capture the full diversity. We also profile the genome level composition for each continent. And we found that the Saccharomyces and the Candida are always the most abundant genera for each continent. And we found that the geography can explain the variation of the human gut microbiome. Also, we found that the European population characterizes the expansion of the Saccharomyces, and the North American population have a higher abundance of the Candida. And lastly, the Asian population contains a higher abundance of the Aspergillus. And we also found that the fungal diversity can differ significantly across the different continents. To further investigate the overall structure of the human gut microbiome, here we use an subsurface clustering method. And here we identify the 4 fungal enterotypes. And here we identify 4 fungal enterotypes in both the ITS1 and SIT2 datasets. And the driven genera of each enterotype are shown here. Here we define the Saccharomyces and the Candida-dominated enterotype as the cyclotype and the can type separately. And the other 2 enterotypes were ASP type and ASC type. And to further evaluate the robustness of the enterotype clustering results, here we randomly removed a certain percentage of the samples and performed recasting. We have found that the recasting result is always stable, and even when after removing half of samples, the less than 5% of samples are wrongly assigned. And we also can find that we also see that these four fungal enterotypes can display significant difference at other taxonomic levels. And we want to see whether the enterotype defined in these two datasets can share structural similarity. So here we performed the cross dataset variation analysis by building a four enterotype classifier. And the four enterotype classifier was trained on one dataset and predicted enterotypes on another one dataset. And we can see that the model can still achieve a high accuracy even after removing the driver general. So this means that the enterotype not only capture the differences in the abundance of the driver general, but also capture the overall structure of the fungal composition. So here we can also see that there are consistent enrichment trends of enterotype-specific fungal general across each study. So this means that the enterotype defined in each study also share high similarity. So we can see that most of the cohorts can contain the 3 or 4 fungal enterotypes. So this means that the enterotype is also independent of geography, but there may be slightly difference in 3 continents. We can see that the second type is more prevalent in the European population, and the kind type is less common in the North American population, where the Asian population contains a higher proportion of the ASP type. And next explore the relationship between the bacterial enterotype and the fungal enterotype. Although we didn't find a one-to-one correspondence between these 2 enterotypes, but we found that there still exists strong correlation between these 2 enterotypes. For example, the can type is still enriched in this E1 bacterial enterotype, and the cycle type is enriched in the E3 bacterial enterotype. And also, the driver general of the bacterial enterotype can show significant differences across different fungal enterotypes. So this indicates that the fungi and the bacteria has close interactions in our human gut. And we next explore the associations between the fungal enterotypes and the host's basic characteristics, such as age. And we found that the age has large factors on the fungal enterotypes. As we can see from this picture, the can type is always enriched in the aged population, and the cycle type is enriched in the young population. Now, we also found that there display significant positive negative correlation between the age and the fungal diversity. And having observed the large effect of age on fungal enterotypes, we next want to identify age-associated fungal genera. Here we use the multivariable linear regression analysis, and a total of 21 age-associated fungal genera are detected. And interestingly, we found that 12 of these genera are also enterotype associated. So this result may explain the age preference among the fungal enterotypes. And then we use this age-associated fungal genera, we calculate a gut aging index GIS score. The GIS score here is used to quantify the balance between the age-positive fungal genera and the age-negative fungal genera, and the higher GIS score will indicate the higher intestinal aging degree. So according to our result, the GIS score within each enterotype shows strong positive correlation with the age. So the GIS score, then we then use the GIS score to validate on another data set that without age metadata. So we want to validate whether other data sets can have the similar age distribution. And we can find that the can type is always has the highest GIS score, where the sec type is always has the lowest GIS score. So this indicates that the can type enterotype contain more age-positive associated fungal genera than the age-negative fungal genera. And so the can type is associated with a higher intestinal aging degree, and where the sec type is associated with the younger status. And we also found that the GIS score are linked to increased disease risk. As we can see that the GIS score in disease of patients, in patients of disease always has a higher GIS score compared to the healthy ones. And we also found a positive correlation between the GIS score and the ECOG score. The ECOG score is an indicator of the function status of cancer patients. So these two results indicates that there may be an overlap between the aging-related and disease-associated alterations in the gut microbiome. And we further explore the clinical relevance of the fungal enterotypes. We can see that the cancer type is always higher prevalent in the diseased patients, where the cycle type and the ASC type is less common in the diseased patients. We also calculate the gut microbiome health index. This index is proposed in a previous study that was used to measure the health status of the human gut microbiome. And similarly, we can find the cancer type has the lowest GMHI index. And finally, we compare the intestinal barrier function across the 4 fungal endotypes by calculating a human DNA contents. This ADC score can measure the intestinal barrier damage degree, and we can find that the cancer type is always associated with the more serious intestinal barrier damage. So this may explain the increased disease risk of the cancer type enterotype. And lastly, we explore the functional variations between these 4 fungal types. By using paired metagenomic samples, we can profile the functional composition of these 4 fungal types. And we identified 31 metabolic pathways that can differ among these 4 enterotypes. We can find that the functional difference between these fungal enterotypes can be partially contributed to the driver general, as we can see that the pathways enriched in the sex type always correlated with the saccharomyces. This is its driver general. And we also found that the function difference across these 4 fungal enterotypes can partially explain the phenotypic differences that we observed before. For example, two heme synthesis pathways were found to be enriched in the cancer type enterotype. And the heme is a key iron source of the pathogenic bacteria and has been previously reported like to have potential damage to the intestinal barrier and that can cause human diseases. And we also found that this pathway, carbohydrate degradation pathway, enriched in the ASC type that shows strong positive correlation with the BMI value, and we found that the ASC type can have a slightly higher BMI values compared to other fungal endotypes. And finally, we found one fungi contributed aerobic respiratory pathway that were enriched in the cancer type can show strong positive correlation with both the AG and the HDC. So we performed the bidirection mediation analysis to explore their relationships. We found that this aerobic respiration pathway can mediate the association between the age and the HDCs. So this means that the increased aging will cause the compromised intestinal barrier by affecting the abundance of this pathway. So in summary, we identified and characterized the 4 fungal endotypes. And we found that these 4 fungal endotypes have a strong age preference. For example, the cancer type is enriched in the adult population, where the sex type is enriched in the young population. And the cancer type can also confer an increased risk of multiple diseases. And lastly, we find that the cancer type associated respiration pathway can mediate the association between the aging and the compromised intestinal barrier. And finally, I will thank to my supervisor, Professor Zhao's guidance, as well as all my collaborators. Thank you, that's all. Thank you for listening.
Time for 2 questions.
Thank you for your interesting talk. I wanted to ask how these enterotypes are decided by the sequencing of ITS sequences in fungal organisms, right?
You mean the sequencing technology for the fungal composition?
Yes. I just wanted to clarify, how are the microbial fungal organisms, like the gut microbiota, especially the fungal microbiota, how are they differentiated from the acquired fungal organisms during hospital infections? as far as I know, in the Asian regions, Candida is the most acquired hospital infections. But we could see in your presentation, the microbiota is Aspergillus.
So, sorry, you mean what? In my study, I use the LTS sequencing net or target specific for fungi. So, and it is amplicate sequencing technology.
Okay, thank you. Thank you so much.
Hi, really great talk. I'm curious whether there's actually any data on like whether the same patients that you studied with all the different fungal types, whether that's like they have cancer data on that as well. Because I think the question is there any data that you can use with colorectal cancer, for example, that you can use to compare the different sort of fungal phenotypes in your study?
You mean the integration of multiple source of samples?
Integration of maybe if you have cancer data, for example?
Actually, our samples include both the healthy samples and the disease sample, and the disease can include multiple diseases such as CRC, IBD, IBS, and AD. But in our studies, we always focus on the whole samples, but we also perform analysis individually for only the healthy samples, but we see that the same conclusion can be obtained.
Okay, thank you so much.